Dear students, in this module, I will introduce open reading frames and the associated concepts to you for the conversion of an RNA sequence into a protein. As you know, codons of 3 nucleotides, they code for each amino acid, and there are multiple amino acids that form the proteins. There is one start codon and 3 stop codons that you may use during this conversion process. And the most important thing that we're going to talk about today is how to search for the start and the stop codons. So this is a very interesting question because this will totally change your protein if you have a different start codon or a different stop codon. For that, the concept of open reading frames needs to be introduced. As you can see in this figure, we have the DNA sequences provided here in the middle of the figure. You can note that the 5' to 3' direction on the upper strand and the 3' to 5' on the lower strand. So the lower strand is obviously complementary to the upper strand. Now, to move towards the open reading frames, so just consider the 5' to the 3' strand. If you have this strand with you and its sequence, then you can have 3 possible open reading frames. Why 3? So because each codon is comprised of 3 nucleotides, so either the beginning of this sequence can be the first, the second, or the third nucleotide. So depending upon which nucleotide you start from, you have 3 different options. If you start from the first nucleotide and form a codon comprising of 1, 2, and 3, then you have the first start codon. But if you choose to form the codon by looking at the second nucleotide, then you will need to have second, third, and the fourth nucleotide, which will form the first codon. You also have a third option. You may choose to start coding your protein from the third nucleotide. In that case, you will have 3, 4, and 5 as the first codon. So therefore, you have 3 options for encoding the first start codon. Why not 4? Because if you have the 6th nucleotide here, then essentially you are saying that 4th, 5th, and 6th nucleotide will form the start codon, but that would mean that the first codon can also be 1, 2, and 3. So therefore, you can have a maximum of 3 possible positions for starting the start codon. You remember I talked about the complementary strand that is given here. So similarly, you can have 3 different start positions for the complementary strand as well. So this would mean you have 3 positions to start from, 3 possible positions to start from in the 5' to 3' sequence, and 3 possible positions to start from the 3' to 5' positions. So in all, you have a total of 6 possibilities. Now, out of this 6 possible starting nucleotides, which one is valid? So the rule of thumb is the longest ORF is valid. In this example, the longest ORF is denoted by the red here. So your ORF number 1 is the longest. Therefore, we will use this to form the protein sequence. This is the rule of thumb which you can remember and you can apply towards encoding your proteins. One can always find web resources for finding the ORF. Instead of doing protein encoding by looking at each individual nucleotide, you can simply paste your sequence at the NCBI's ORF finder, and it will give you the longest possible ORF that exists within your nucleotide sequence. So this will surely simplify your problem. In conclusion, there are 6 ORFs that are possible for any nucleotide sequence, and you need to find out which ORF is the valid one. The rule of thumb is that the longest ORF is the one that you have to work with. And of course, the first stop codon that comes once you enter the reading frame will mark the end of your protein.